Today, we're diving into a fascinating topic bridging chemistry, evolutionary biology, and the origins of life, mapping the chronological order in which amino acids appeared and were incorporated into the genetic code. What amino acids came first? What amino acids appeared later? This has important implications for our understanding of the origins of life. Now, there's multiple criteria that we can apply to determine this chronology. Later today, my groupmates will discuss other ways that may help us reach closer to the truth. But today, right now, the hypothesis that I'm exploring today is that simpler amino acids, those with lower molecular complexity, were more likely to have formed under the conditions of early Earth, and thus appeared earlier in the timeline of prebiotic chemistry. If we consider this hypothesis, then amino acids with simpler structures were amongst the first to appear. Take a look at this timeline. This is our current understanding of some of the amino acids and the sequence in which they appeared in. You can see in the leftmost here, glycine. It has a very simple structure and very few bonds. So that may be a clue that it appeared earlier. And then valine, which is next in the timeline, is considered to have appeared after glycine. You can see in the structure that it has a few more bonds, and its structure isn't as simple as glycine's. In the middle of this timeline, we have proline, which is one of the 20 amino acids, and it has a ring, which is considered a sign of molecular complexity. But what is molecular complexity? For a long time, scientists just treated it on a I know it when I see it basis. For example, which of these amino acids looks more complex, glycine on the left or tryptophan on the right? And I think intuitively, most of us will say that tryptophan is more complex than glycine because it has two ring structures. And that would be the scientific consensus today. But it's not always that clear cut. Between phenylalanine on the left and methionine on the right, which one do you think is more complex? Well, some scientists may say that phenylalanine is more complex because it has a ring structure, while other scientists may say that methionine is more complex because it has a sulfur atom, which means it has more chemical diversity. Here, the need for a structured, methodical approach arises. How do we define and quantify molecular complexity? Different scientists have come up with different methods to quantify this. There have been at least 23 distinct methods for quantification, but they can be broadly categorized into structural, topographical, and graphical. Let's first focus on structural methods of quantifying how complex a molecule is. Structural methods are those that focus on these structural characteristics of a molecule. Earlier, I mentioned the rings of tryptophan. I was taking a structural approach to quantifying its molecular complexity. So one example of a method that focuses on the structural characteristics of a molecule to determine how complex it is called the normalized spatial score. It considers criteria such as atom hybridization, stereoisomerism, the presence of a non-aromatic ring, and the number of heavy atom neighbors. But what do these terms even mean? Well, atom hybridization is when atomic orbitals mix to form new hybrid orbitals, which affects the molecule's geometry. Stereoisomerism is the phenomena when molecules have the same molecular formula, but different spatial arrangement. And what are heavy atoms? Well, heavy atoms are any atoms that are not hydrogen. Now, between aspartic acid on the left and proline on the right, these are two amino acids. But which one of them do you think is more complex? Well, earlier, I just said that that one of the criteria that the normalized spatial score considers is a non-aromatic ring, and proline has that characteristic. So according to this method of quantifying molecular complexity, proline would have a higher score. It would be considered more complex than aspartic acid. Another method to quantify molecular complexity is assembly theory. The central idea here is how trying to quantify the minimum number of steps that you can use to build any entity from its most basic building blocks. So consider these red squares on the left here. These are simple bonds. They are considered the most fundamental building block of any molecule. You combine them together to build more complex structures, which you combine together to build even more complex structures. And assembly theory is the method of assigning each molecule a minimum number of steps that you would need to achieve it. Now, according to assembly theory, which amino acid would have a higher score? Arginine on the left or cysteine on the right? Now, visually, I think we can all see that arginine would require more steps to assemble it compared to cysteine. And arginine is indeed the amino acid that would have a higher assembly theory index. Another type of approach to quantifying molecular complexity is topographical. Topographical approaches are those that focus on connectivity and the spatial arrangement of atoms. One example is the Proudfoot CM index. It considers criteria such as types of paths from each molecule and the total number of paths in this molecule. But what does that even mean? Well, consider this carbon atom in this amino acid right here. It has one path emanating from it, a carbon-carbon bond, and another path emanating from it, the carbon-nitrogen bond. These are two chemically distinct paths. And then that is one of the values that you would plug into a complicated mathematical formula that I won't be covering in this presentation. And the other value that it considers is the total number of paths in this entire molecule. So all of these paths here. Now, between valine on the left and histidine on the right, which amino acid do you think would be considered more complex according to the Proudfoot CM index? Well, visually, we can all see that histidine seems to have a larger number of paths that comprise this molecule, and it is indeed the amino acid that has a higher Proudfoot CM index score. Another type of topographical method of quantifying molecular complexity is the Botcher score. It considers different criteria such as the total number of bonds, the number of chemically non-equivalent bonds, the number of different isotopes, and more. Consider this carbon atom that's been highlighted by the red dot. The total number of bonds here are 3. It has the number of isomeric possibilities at this position is 2. It has 4 valence electrons, and so on. You would plug all of these values into a mathematical formula that Botcher provided, and it spits out a certain value that you can use to compare how complex different molecules are from each other. So consider these two amino acids, histidine on the left and phenylalanine on the right. Now, both of them have rings. So which one do you think would have a higher Botcher score? Well, one of the criteria that this considers is the number of chemically non-equivalent bonds. Here, you can see that the ring in histidine has 2 nitrogen atoms in it. So it has less chemical equivalence than the ring in phenylalanine, which seems somewhat uniform. Now, the last method of quantifying molecular complexity is graphical. Graphical approaches are those that utilize graph theory, which is a subfield of mathematics. One example is minimal graph theory. So here on the right, you can see the structure of cysteine, which is an amino acid. Through some complicated graph theory, you can convert this structure into a linear sequence of symbols. And then the length of this sequence can be a measure of how complicated it is using some more mathematics. Now, I've been talking about different methods of approaching and quantifying how complicated a molecule is. But is there any consensus on which molecules are consistently rated the most complex and which amino acids are consistently rated the least complex? And it turns out, yes, there is. So here highlighted with the red squares, you can see that glycine has a very simple structure compared to the other amino acid structures that you can see. And in every single method, it is considered to be the most simple amino acid. Other amino acids that are consistently rated very, very simple are alanine and valine, highlighted with the red squares over here. Visually, you can see that they have relatively simpler structures compared to the other amino acids that you can see on the screen. What amino acids are consistently rated to be the most complex? The answers are tyrosine and tryptophan. Here you can see these ring structures, which are a mark of molecular complexity, make them one of the criteria that make them one of the most complex amino acids that are present in our world. Now, I would like to start off with a note. I would like to use a note here that we cannot simply assume that just because an amino acid is the simplest, then that necessarily means that it's the first amino acid to appear. We have to integrate different criteria that my group mates will be discussing later today, such as the biochemistry, meteorite data, experiments that try to recreate Earth's earlier conditions. We have to integrate all of these criteria together to determine which amino acids came first and which amino acids came later on. Now, what is the significance of determining with the chronology of amino acids and the order in which they appeared? Well, one of them is primitive life forms. Uh, if we know which amino acids came first, it can help us understand how the earliest life forms, the most primitive ones, how they processed energy and nutrients. Another point of significance is that it can give us a deeper understanding of the origins of life. Earlier amino acids were the ones that interacted with RNA, which led to certain biological processes, such as translating genetic information. And lastly, if we understand amino acids and chronology, then we can also understand protein development, or at least part of it. And to summarize my presentation, there are different methods of measuring how complex a molecule is. If we apply these complexity measures to amino acids, we can understand which amino acids came first and which amino acids came later. But molecular complexity is not the only criteria that we should be using. We have other criteria that my group mates are using, such as meteorite data and experimental data. Thank you.
Yeah, that was awesome, Ameena. If
Thank you.
others have questions out there, reminder, you can ask questions in the chat, or you can raise your hand and I'll ask questions for you or call on you. I see Tomasz has his hand up.
Hello. Hi. First of all, amazing talk, so clear. And I'm going to be sharing the recording with all my friends because we discuss about how primordial the amino acid can be. We actually work with peptides, prebiotic peptides, so it super concerns us if the peptide is made of amino acid that we consider prebiotic or not. And I want to share one comment from Andrew Pohorile that I heard and stuck in my head as an interesting concept, and then a short question. So the comment was that he said, Tomasz, if you find a peptide that is incredibly useful from the perspective of the origin of life, but it consists mostly of amino acids that we think are not prebiotic, there will be enough research to find out that they're actually prebiotic. So kind of a joke, but you see, it just shows us like how fragile our knowledge is and how little we explored with the possible pathways to synthesize them. So something to keep in mind that if something is not necessarily considered prebiotic, it's still probably worth working with those peptides still. That's for the peptide people. Yeah, super useful to have those parameters for complexity. I like that you use them for amino acids. It's probably interesting to see in general how complexity. I'm not sure like how far in a scale of complexity we can use those kind of approach, like for macromolecules or not. But that's not even my question. My major question is, do you know? It's okay if you don't. It's just a literature search that I can do. What is like a surprising amino acid that was found on meteorite that we wouldn't expect it to be prebiotic, and maybe it is?
Okay, so my group mate Jonathan is going to be doing the meteorite part of this project,
Perfect.
so I think it's better to ask him.
I'll wait for his presentation then. And once again, congratulations. Thank you for a beautiful talk.
Thank you.